You ever wonder about that? I mean, we all know things like heart disease or asthma can run in families, right? So you'd think it would be easy to just find the gene responsible. But for the longest time, it's been this huge scientific mystery. We knew genetics were a major player, but finding the actual culprits, way harder than anyone expected.
Well, it turns out the answer is buried in this incredible statistical detective story, and that's what we're going to dive into. We're going to follow the clues, we're going to expose the false leads, and we're going to uncover the amazing forensic tools that scientists had to invent to crack the genetic code of common diseases.
So here's our game plan for this investigation. We'll kick things off with a totally new way of hunting for genes. Then we'll check out the first toolkit scientists used and see why it had some serious flaws. Next up, the breakthrough model that changed everything, and finally, how we go from just finding clues to actually proving who the culprit is.
Okay, so let's get into it. Our story really starts with a complete change in how scientists even thought about looking for these genetic clues. It was a huge shift.
See, the old way of doing things, called linkage analysis, was all about studying big family trees. And you know what? It worked great for these super rare diseases caused by one single powerful rogue gene. But for common stuff like diabetes or asthma, it just didn't work. Scientists started to realize that these diseases aren't caused by one big genetic hit, but by maybe hundreds or thousands of common genetic variants, each one having just a tiny little effect. And to find those, they needed a whole new weapon, the Genome-Wide Association Study, or as everyone calls it, GWAS. This new method scans the DNA of thousands of totally unrelated people all at once.
So what exactly are they hunting for in all that data? Well, the main clue is something called a SNP. That's short for single nucleotide polymorphism. The easiest way to think about it is like this. Imagine your DNA is a 3 billion letter instruction manual. A SNP is just a one-letter typo. These little variations are super common, and they act like millions of molecular breadcrumbs that our genetic detectives can follow. Now, this massive hunt didn't just happen overnight. It was only possible because of a few huge breakthroughs. First, in the early 2000s, the HapMap project gave us a reference map, a catalog of all these common SNPs. Then, new tech called SNP arrays came along, which let labs scan for hundreds of thousands of these clues super fast and cheap. And by 2005, the first big GWAS paper was published, and it was a smash hit. It proved this new approach was the real deal. Okay, so the hunt is officially on. The technology is there. Now let's open up the toolkit and see what kind of statistical tools these genetic detectives were working with in the beginning.
In the early days, the workhorses were some pretty classic statistical tests. It didn't matter if you were looking at something binary, like case versus control, do you have the disease or not, or something quantitative like your height. The basic idea was the same. You take millions of SNPs and you test them, one by one, using these fast, simple models.
And when you run literally millions of these tests and plot the results, well, you end up with one of the most iconic images in all of genetics. It's called a Manhattan plot, and you can see why. It really does look like a city skyline. Each one of those little dots is a single SNP, and how high it is on the plot tells you how strongly it's linked to the disease we're studying. And those really tall skyscrapers, those are your hot leads. They're pointing to the genetic neighborhoods that scientists need to investigate right away.
But, every good investigation needs a lie detector, right? For GWAS, that's something called the QQ plot. It's a way to check if your results are legit. If everything is perfect, you get a nice, clean, straight diagonal line. But in the early studies, scientists kept seeing this weird upward curve, and that was a major red flag. It was basically screaming that the evidence was contaminated with a ton of false positives.
And that brings us to a huge twist in our story. The first toolkit was definitely powerful, but it was being tricked. The evidence was leading detectives astray because of some very clever problems hiding in plain sight.
The number one villain here was something called population stratification. Okay, imagine this. A certain SNP is just more common in one ancestral group, say people of European descent. And for totally different reasons, like diet or environment, a particular disease is also more common in that group. Well, a simple statistical test will scream, aha, this SNP is linked to the disease. But it's a complete illusion. It's a classic false lead created by ancestry, not biology.
And the second bad guy was something called cryptic relatedness. A core assumption of GWAS is that everyone in the study is unrelated. But a lot of the time, they aren't. You might have people who are third or fourth cousins and they don't even know it. Their shared DNA messes up the math and can make a statistical signal look way stronger than it actually is, which just leads to more false alarms.
All of these problems led to the biggest mystery of the era, the case of the missing heritability. Look, scientists knew from studying twins that traits like height are about 80% genetic. But the first big GWAS studies, even after finding dozens of SNPs, could only explain about 5% of that. So where was the other 75%? It was like the vast majority of the genetic story had just vanished into thin air.
And we're talking a massive gap here, almost a 10-fold difference. This was the key clue that something was wrong with the methods. The heritability wasn't biologically missing, it was statistically hidden. The models just weren't sufficiently enough to see the combined effect of thousands of genes, each with a teeny-tiny influence.
So this is the turning point in our detective story. Scientists were facing false leads, contaminated evidence, and a huge chunk of the puzzle was just missing. They needed a breakthrough, and they got one. The solution was a revolutionary new kind of statistical model called a linear mixed model, or LMM. And the genius of these models was that they didn't just try to put a band-aid on the old system. Instead, they created a single, powerful framework that could solve both problems, population structure and cryptic relatedness, at the same time.
So how on earth does it work? Well, the idea is actually super elegant. First, instead of assuming everyone is unrelated, the model calculates the exact genetic similarity between every single pair of people in the study. Then, it bakes that information right into the statistical test for each SNP. It essentially allows the model to see the background genetic noise from ancestry and family ties, and cleanly separate it from the true biological effect of the SNP it's testing. It's brilliant.
And this wasn't just a theory. This breakthrough was quickly turned into powerful software that researchers could actually use. Tools like GCTA were the first to really solve that missing heritability problem, and later, programs like Bolt LMM made the process so incredibly fast that they can run these super complex analyses on massive datasets, like the UK Biobank with 500,000 people. The modern genetic detective suddenly had a very, very high-tech toolkit. All right, so this brings us to the modern era. Now that we can get clean, reliable evidence, the goal of the investigation has shifted. It's no longer just about finding a link, a correlation. Now, it's all about proving causation. See, a skyscraper on a Manhattan plot doesn't actually point to one single genetic culprit. It points to a whole neighborhood of suspicious SNPs that are all correlated with each other. So the next step is a statistical technique called fine mapping. Think of it like bringing in a whole lineup of suspects and using sophisticated analysis to figure out which one is the most likely to be the actual bad guy. The result is a credible set of the top suspects. But finding the culprit isn't enough, right? A good detective wants to know the motive. Why did they do it? To figure out why a SNP causes a disease, scientists now use something called multi-omics integration. They take the genetic clue, the SNP, and they link it to other layers of biological data, like which genes are being turned on or off, what proteins are being made, or what metabolic pathways are being affected. It's about building a complete story, from that one little DNA typo all the way to the disease itself.
So this incredible, decades-long statistical journey has finally given us the tools to find the real genetic culprits behind so many common diseases. And it leaves us with this one profound question for the future. Now that we know how to find the cause, how long will it be until we can predict, and maybe even prevent, the crime from ever happening in the first place?